Hi, I'm Zachary Pagan, and I'm here to talk about GMO foods. GMO foods are not a new concept. However, they are feared by the public, and I'm here to debunk some myths about them. I'm going to go through the process on how GMOs are created and look at their overall potential. Gene modification has been going on for years. Crossbreeding is a similar but less effective process that has been done for a long period of time. In order to crossbreed, pollen is taken from the stamen, or male organ of one plant, and is transferred to the pistil, or female organ of another plant. The purpose of this is to exchange genes between two plants. However, it is way more inefficient when compared to GMOs. The purpose of this was to create superior plant varieties, and GMOs are no different. Despite the long history of gene editing, only 57% of the general public believes that GMOs are safe to consume, and this can likely be attributed to misconceptions, such as the idea that GMOs can mutate humans and that they are abnormally large. In fact, 88% of scientists from the American Association for the Advancement of Science, or AAAS, believe that GMOs are in no way harmful. The general public must come to the realization that GMOs are produced for a practical purpose, and that purpose is to increase crop yield, produce superior plant varieties, and to create more nutritious animal feed. This would help us feed the exponentially growing population and overall revolutionize the agricultural industry. There are two types of GMOs, and the first type is our first generation GMOs, which have herbicide resistance and insect tolerance. This reduces food protection costs for farmers, which would help the struggling farmers. The second kind of GMOs are second generation GMOs, and these have nutritional or industrial traits that could help reduce obesity. Non-browning apples are a second generation GMO that are quite successful. Make sure to not get caught up in all of the misconceptions. Gene editing is a complex process that starts with isolating the gene by comparing two different species that possess the gene and finding where they have the same DNA using a process called comparative analysis. Next, the DNA is cut and pasted onto a plasmid and inserted into the Agrobacterium tumefaciens. Finally, the bacteria invade the seeds of the plants and insert their DNA into the plant, which genetically modifies it. Another method is to coat the DNA on gold or tungsten and then shoot it with a gene gun. After the gene is inserted, the plants are grown in expensive climate-controlled chambers. Once the plants have grown properly, the GMO has been fully developed and can be grown on a larger scale. As discussed earlier, GMOs are capable of helping us achieve many great feats. However, there are risks such as the modified gene escaping into the wild, insects evolving to overcome insect-resistant crops, or allergens being introduced into our food, which would be catastrophic. Due to these risks, we must proceed cautiously when producing GMOs, and there's no telling what the future may hold.